Welcome to this tutorial. Here we are going to explore the membrane proteins.
But before that, don't forget to subscribe the channel and click on the bell icon to get all new notifications and keep up to date about all the new uploaded videos in this channel.
You know, phospholipids form the basic structure of cell membrane and make it semi-permeable. They also help in compartmentalization in cell. But, the most active functions of the membrane are carried out by the membrane proteins like selective transport of molecules and nutrients, passage of water, maintenance of proper ionic concentration inside the cell, and many more like cell adhesion, enzyme activity, expression of cell identity, cell recognition, energy transduction, cellular signaling, and so on. At the very first step of exploring membrane proteins, we have to have some idea about those proteins. So come on, let's discuss about that. The proportions of proteins in the plasma membrane vary between different cell types and species. In animal cells, about 30% of the proteins are membrane proteins. In general, membranes which are actively involved in metabolism, contain a higher proportion of protein. The membranes of organelles, like mitochondria, chloroplast, which are actively associated with energy transduction, have about 75% membrane proteins. Myelin sheath, which is a lipid-rich membrane, insulates nerve fiber, and contains only about 20% proteins of its dry mass. Proteins are associated in the membrane in several different ways. Like lipids, proteins can also diffuse laterally in the membrane, but at very slow a rate. Their orientation in the membrane are in asymmetric manner. This is the main cause of membrane asymmetry. Membrane proteins are categorized on the basis of their structure, position in the membrane, and function. These proteins are classified mainly into three categories depending on their association with the membrane. They are intrinsic or integral membrane proteins, extrinsic or peripheral membrane proteins, and lipid-anchored proteins. Now we are discussing about these three types of protein. So number 1. Integral proteins. Integral membrane proteins penetrate the cell membrane, and are permanently attached to the membrane from one side. Their non-polar regions are firmly embedded in the hydrophobic region of the lipid bilayer, whereas the polar regions of the protein protrude from the lipid bilayer. Almost all intrinsic proteins contain special amino acid sequences, generally about 20 to 24 amino acids long, that extend through the internal regions of the cell membrane. Such proteins can only be separated from the membranes by using non-polar solvents, detergents, such as SDS, or Triton X100, or maybe by using some denaturing agents. Integral membrane proteins include transporters, linkers, channels, receptors, enzymes, structural membrane anchoring domains, proteins involved in accumulation and energy transduction, and proteins responsible for cell adhesion. Some of the examples of integral membrane proteins are rhodopsin, glycophorin, glucose permease, porins, ion channels and gates, insulin receptor, cell adhesion proteins like adherins, selectins, etc., gap junction proteins, G-protein coupled receptors, etc. Integral membrane proteins can be classified into two groups. A. Transmembrane proteins. They are the most common type of integral membrane proteins. They span the entire membrane. In the following diagram, number 1, 2, and 3 are transmembrane proteins. B. Integral monotopic proteins. They are also permanently attached to the membrane but only from one side, that is, they embed into a single face of the membrane. Number 4, 5, 6, and 7 in this diagram are the integral monotopic proteins. A. Transmembrane proteins. The most common type of integral membrane proteins are transmembrane proteins. They span the entire membrane. There are different types of transmembrane proteins based on their structure, and how they span across the membrane. Such as, A. Single-pass transmembrane proteins, that cross the membrane only once. B. Multi-pass transmembrane proteins that weave in and out, crossing several times. A. Single-pass transmembrane proteins. They are alpha helix proteins that cross the membrane only once, that is, they are bitopic proteins. This type of protein usually has: number 1. A hydrophilic section containing charged and polar amino acids in the aqueous environment outside the cell. Number 2. A hydrophobic stretch of 20 to 25 nonpolar amino acids, forming an alpha helix within the hydrophobic core of the bilayer. So only the alpha helical segment of the protein crosses the bilayer. And number 3. A hydrophilic portion within the aqueous interior of the cell. Glycophorin A is an example of single-pass transmembrane proteins. There are again two types of single-pass transmembrane proteins. Type 1. Their carboxyl termini is towards the cytosol, and amino termini is towards the extracellular space. Type 2. Their amino termini is towards the cytosol, and carboxyl termini is towards the extracellular space. So it is just the opposite of type 1. B. Multi-pass transmembrane proteins. They can be alpha helical proteins, or beta sheet proteins or beta barrels. Beta barrels are less common. Both of them cross the membrane multiple times. So they are polytopic proteins. Transporters and ion channels are examples of multi-pass transmembrane proteins. Bacteriorhodopsin and many cell surface receptors are the examples of alpha helical proteins. Whereas, porin, which is a transporter protein, is an example of beta barrel proteins. The core of this transporter protein generally is hydrophilic, permitting passage of water-soluble molecules. Multi-pass transmembrane proteins also have some subtypes. Type 3. They have multiple transmembrane domains in a single polypeptide chain. Whereas, type 4 proteins consist of several different polypeptides assembled together in a channel through the membrane. There are also some subtypes of transmembrane proteins. Type 5. They are anchored to the lipid bilayer through covalently linked lipids. And type 6, they have both transmembrane domains and lipid anchors. B. Integral monotopic proteins. Unlike traditional transmembrane proteins, monotopic membrane proteins enter and exit on a single face of the membrane, as opposed to completely spanning the membrane bilayer. Monotopic membrane proteins expose all soluble domains towards only one side of the membrane. Right side of this schematic representation is showing different types of interactions between monotopic membrane proteins and the cell membrane. Number 4 in this diagram is denoting the interaction by an amphipathic alpha helix, parallel to the membrane plane. Number 5 is showing the interaction by a hydrophobic loop, or hairpin. Number 6 is the interaction by a covalently bound membrane lipid. And number 7 is the ionic, or electrostatic interactions, with membrane lipids. Now number 2. Peripheral membrane proteins. Peripheral proteins are found on the surface of the membrane on both cytoplasmic side and extracellular side of cell membrane. They interact with the membrane less extensively. Most of the peripheral membrane proteins are hydrophilic. Unlike integral membrane proteins, they don't enter into the hydrophobic region of the cell membrane. They attach to integral membrane proteins or penetrate the peripheral regions of the lipid bilayer. They attach to either the polar heads of phospholipids, or onto the polar region of an integral protein, or another peripheral protein. These bonds are typically hydrogen bonds, or other non-covalent interactions, like ionic bonds. This feature helps these proteins to work only on the surface of the cell membrane. Peripheral proteins can be activated, or disabled through a number of different pathways. Many peripheral proteins are also a part of many complex biochemical pathways. Addition of a salt solution, or changing the pH, can disrupt those weaker bonds, and dissociate peripheral proteins without disrupting the integrity of the membrane. Peripheral membrane proteins have a lot of important functions. These proteins can be involved in moving substances, or transportation, within or outside of a cell, activate other proteins and enzymes, or be involved in cell-to-cell interactions. Some peripheral proteins maintain and support the intracellular cytoskeleton. They give a cell its shape, offers support, and facilitates movement through three main components: microfilaments, intermediate filaments, and microtubules. They also direct many components of the extracellular matrix. Some peripheral proteins help in communication. They receive signal from outside the cell, and then pass the signal to the next protein, integral protein, or another peripheral protein it attached to. The message is then carried inside the cell. Some serves as receptors, surface antigens. Several peripheral proteins work as enzymes to break down specific substrate, or to combine it with another molecule. Some of them control the synthesis and destruction of the cell membrane itself. Many peripheral proteins are also involved in transferring small molecules or electrons. Many of such proteins are found within the electron transport chain as electron carriers. These proteins transfer electrons from integral proteins they are attached to, and can pass the electrons to other proteins and molecules. There are some hydrophobic molecules which bind to some peripheral proteins, and are passed through various methods across, or through the membrane. Some peripheral proteins function as carriers of nonpolar compounds between different types of cell membranes, or between membranes and cytosolic protein complexes. They serve as regulator for membrane-bound enzymes, also may limit the mobility of integral proteins. Some examples of peripheral membrane proteins are ankyrin, spectrin. Proteins involved in electron transport chains, such as cytochrome C, as shown in the following diagram, Q-redoxins, high-potential iron protein, adrenodoxin reductase, some flavor proteins, and others. Now number 3. Lipid anchored proteins. In eukaryotic cells, some integral membrane proteins don't enter the lipid bilayer, rather is anchored in one leaflet by covalently attached fatty acids or phospholipids. Actually, some integral and peripheral proteins may be post-translationally modified, with added fatty acid, for example, palmitate, or myristate, diacylglycerol, or prenyl chains, or glycosyl phosphatidylenoacetyl or GPI, which may be anchored in the lipid bilayer. Now they are called lipid-anchored proteins. Here I'm showing how does a lipid-anchored protein form. You can see here a covalent bonding between a lipid group and a peptide chain that is nothing but a protein by the process called lipidation. And this structure helps the protein to be anchored in one leaflet of the membrane. Now you can better understand it by viewing this animation, which is showing a protein that is post-translationally modified and works as an anchor, which is finally embedded or anchored in one side of the lipid bilayer. The lipid-anchored proteins can be located on either side of the cell membrane. On the cytoplasmic side of the cell membrane, lipid-anchored proteins are covalently attached to different fatty acid acyl chains. Whereas, on the opposite side of the cell membrane, that is at the cell surface, lipid-anchored proteins are covalently attached to the lipids, glycosyl phosphatidylenoacetyl or GPI, and cholesterol. This structure, by which the protein itself remains outside the bilayer, but firmly connected to the bilayer by its phospholipids, gives these proteins a relatively high mobility compared with other proteins. There are also some other types of proteins based on their transport mechanisms. Let's see. So, according to the transport mechanism through the membrane, there are two extremely important types of integral proteins that are found all over the cell membrane, and help the cell maintain homeostasis or balance. They are, number 1, channel proteins, and number 2, carrier proteins. Number 1. Channel proteins. They allow molecules or small ions to pass through the cell membrane. There is a channel or hole inside the protein that lets things to pass through. For example, sodium ions, which wouldn't able to pass through by themselves from outside to inside. Channel proteins can also do the reverse. When cell has too much sodium ions, these proteins start pumping these out from the cell. Generally they don't require energy, and it goes with the concentration gradient. Number 2. Carrier proteins. Carrier proteins carry substances into the cell, and protect the substances so that it can enter the cell safely. Unlike channel proteins, they can go against the concentration gradient. So carrier proteins can sometimes use energy or ATP. It happens if a cell has a lot of chloride ions, and it needs more to perform a certain work. Like channel proteins, it can also do the reverse, that is pump them outside the cell. So we have explored all about the membrane proteins. Now I just wanna gist it out and conclude it, and you also have to revise this. You know, membrane proteins are very common as well as important proteins. They interact with the biological membranes. They make about 1/3 of all human proteins. They serve as the targets for many drugs. They help in cell communication. And they also might participate in facilitated or active transport mechanism. This concludes the tutorial here. So thank you very much guys for watching this video. You may also wanna see my next video in the models of plasma membrane. You may find some other interesting videos in this channel as well.